in eukaryotes, but not in prokaryote. Because in prokaryote, there is no unnecessary genes, introns. And the gene is very simple. That's why no need to modification in prokaryote. But in eukaryote, need to be modified. That means remove the introns, and add the 5-methyl cap, polyadenine tail. These are the modifications. So let's see the PPT slide. Are you able to see? Hello? Are you able to see PPT slide? Hello?
Yes, sir.
Is it clear?
Yes, sir. Clear, sir.
Right. So yesterday we discussed about how the mRNA prepared from the DNA strand. You can see in this diagram, this is the blue colors DNA strand. And green color is that is mRNA, which is prepared from the DNA strand, right, with help of the polymerase 2 enzyme. Let's take marker. With help of polymerase 2 enzyme, yesterday we have seen how this mRNA prepared, right? While the transcription start, immediately after, actually in this both DNA strands, one is act as a template. One is act as a template. The template strand should be 3' towards 5', because always newly synthesized DNA strand always towards the 5' to 3'. If you see the strands always anti-parallel direction. For example, if you see the DNA strand, this is DNA double helix, one is 5', automatically end with 3', and the opposite anti-parallel direction. That means this side 3' and here 5'. So this is the direction of the DNA. If mRNA prepared a transcription process, this mRNA, green color mRNA prepared like this. But in this, both the template stand is this one, 3 and 2. 3 and 2, 5 and is the template stand. Now, automatically the opposite side, this end is should be 5'. This is 5', and this is 3'. So like this mRNA prepared with help of transcription factors and with help of RNA polymerase 2. Then we will see what happened after mRNA prepared. So the majorly 3 aspects are there. Actually, this first formed RNA, that is called pre-mature RNA or hnRNA. It need to be matured. To be matured, here the requirement from 5 and capping, that is the first one. Second, 3 and polyadenylation and splicing mechanism. These 3 are important. So here the 5' cap formation, 5-side cap formation. Here the polyadenine tail formation at 3'. And you can observe here in this mRNA stand, now when there, apart from the green, another color also observed, another brown color also observed. Is it visible or not? Hello?
Yes, sir.
Right. This brown color, except the green, the remaining brown color, that is called as introns. Introns, they are inactive, not code for any amino acids. They need to be eliminated. So in third step, what happened? These introns are removed. Here, these introns are removed like this. So this removal of introns called as splicing. This is called as splicing, right? So in first what happened? DNA strand transcribed. DNA strand transcribed. DNA strand transcribed. The what it transcribed? mRNA. Then 5 and capped with modified nucleotide. 5 and capped with modified nucleotide. Polyadenine formed. Next, introns removed as well as exons joined. Introns removed as well as exons joined. Now it formed mature RNA. Now the mature RNA transported into the cytosol. Now where is transcription occur? And eukaryotic transcription takes place in, hello? Yesterday we completed transcription process.
Yes, sir.
Where the transcription process takes place. What is the substrate of the transcription? First of all, what is transcription? What is transcription?
Sir, it
Transcription mean
providing DNA
right from DNA strand RNA prepared. Where is DNA? Where is DNA?
Nucleus.
Right, nucleus. Actually, of course, nucleus, but exactly in chromatin material, chromosome, which are present inside the nucleus.
Yes, sir.
Now tell me the where the transcription takes place. In which part of the cell? My voice is clear.
Yes, sir.
Now tell me, definition clear. Transcription means mRNA prepared from the DNA strand. That is first. Second question is, where is DNA? You told that the DNA present inside the nucleus is part of the chromatin material chromosome. Now tell me, that the entire mechanism, transcription mechanism takes place in
Nucleosome.
that's it, nucleus. During transcription process, while preparing the mRNA strand, there need to be post-transcriptional changes. So what are the post-transcriptional changes? What are the 3 post-transcriptional changes? Three post-transcriptional changes already given. 5' capping, 3' polyadenylation, and splicing. These 3 post-transcriptional changes. Okay? Hello?
Okay, sir.
Now these changes also takes place inside the nucleus. These three takes place inside the nucleus. Next what happened? Matured mRNA prepared. Matured mRNA. This mature mRNA now transported into the cytosol. If you take the cell, nucleus, this is nucleus. What is the remaining part? Nucleus, this is nucleus. After nucleus, what is the remaining part? This portion.
Nucleosomes.
I'm asking, are you able to see the marker?
Yes, sir.
This region, I'm asking this region. This is complete cell. This is the cell structure, cell wall, and inside the nucleus, this is nucleus. Now what is between? And just below the cell wall, plasma membrane also present. That also I would have drawn plasma membrane. Now, what is present between nucleus and plasma membrane? This gap,
Cytoplasm.
right? Cytoplasm. Cytoplasm. What are present in cytoplasm? What is present in cytoplasm? What are in cell component except the nucleus? What are the remaining cell organelles? Hello? Cell, what are the cell organelles? I am asking simple question. Cell organelles. What are the cell organelles?
Mitochondria.
Right,
Cytoplasm.
mitochondria, Golgi complex, these are all.
Answer. Yes,
So
sir.
these are all together called as cytoplasm. If you remove the cell organelles, only the empty what liquid, that is called cytosol. That is called cytosol. Cytosol. So now this is this, for example, this nucleus, no cell organelles. Now this is called as cytosol. Now the mRNA already prepared inside the, let's take color, prepared inside the nucleus. Now it is transported into the, after mature, transferred into the cytosol. That's it. What happened after transported into cytosol? Now what is the next stage? After transcription, another stage in central dogma. What are the two stages? Transcription, and second one is, hello? Second one is translation. Now translation takes place inside the cytosol, right? Is it clear?
Yes, sir.
Right. So now let's discuss one by one. First, 5' capping. Next, polyadenine, splicing, right? So these are the post transcriptional changes. Nucleotide recognition. Right. 5' capping. First of all, in 5' capping, remember the three important aspects. These are mainly involved in three enzymes. Actually, involved in mainly three enzymes to formation 5' capping. 5 prime. So first of all, at the end of the transcription, the 5 prime end, let's take the 5 prime end contains free triphosphate group. 5 phosphate group, for example, after formation mRNA strand, are you able to see the marker? Hello? Hello?
Yes, sir. Yes, sir.
Right, right. This is 5 prime and this is 3 prime and 3 prime have free triphosphate. Free triphosphate, right? Since it was first incorporated nucleotide in the chain, the capping process the capping process replaces the triphosphate group with another structure called cap. So now the cap is replaced with the 3-phosphate. Let's see how it is replaced. The cap is added by enzyme guanyl transferases. This enzyme catalyzes the reaction between the 5' end of the tRNA transcript and guanine triphosphate GTP, guanine triphosphate molecules. The guanine transferase enzyme will cleave 2-phosphate bond from guanosine residue and form the unusual 5' triphosphate bond with 5'. Actually, what happened before that? Before involvement of enzyme guanylate triphosphate, the another enzyme required, that is nucleotide triphosphatase. Nucleotide triphosphatase. First of all, this is the first step. Remember. What happened in second step? Same 5.3 point. Same triphosphate. But here what happened before guanosine triphosphate? What happened here? With help of the enzyme, that enzyme nucleotide triphosphatase, nucleotide triphosphatase, nucleotide triphosphatase, which cleave the one phosphate, Now in triphosphate, one is removed. One phosphate is removed. Now it has become only 2-phosphate. And remaining as it is. That means and 5 and 2, 3 and 3 and. Now what happened? Please add this one. Please add second step. Now the cap is added by enzyme guanylate triphosphate. Now the enzyme with help of guanylate triphosphate, enzyme guanylate transferase catalyze the reaction between the 5' transcript and a guanosine triphosphate molecule. The guanylate guanylate transferase enzyme will cleave 2-phosphate bond from the guanosine residue. Right. What happened here? This is already 2-phosphate here. GTP, guanosine, GTP means triphosphate, guanosine triphosphate. In this triphosphate, guanyl transferase enzyme will cleave the 2-phosphate bond from the guanosine residue, will cleave 2-phosphate again here also. Sorry, not a T. Phosphate. In guanosine triphosphate, how many phosphate there? Hello? In guanosine triphosphate, GTP, 3 phosphate. In this 3, with help of the guanylate transferases, 2 cleaved from the remaining one. That means these 2 phosphates separated. Then how many remain? In this 3?
One.
After
One.
removing 2, 1. Now this remaining 1 phosphate, guanosine now it is called monophosphate, right? Hello?
Yes, sir.
Now it is called monophosphate. After removing 2-phosphate, now it is called monophosphate. Now it is monophosphate attached to the 5' 3' chain mRNA. Already the mRNA, 2 phosphates are there, right? 2 phosphate there with 5'. Now another phosphate, guanosine monophosphate, guanine monophosphate attached. Guanosine monophosphate attached. Right? Is it clear? Hello?
Yes, sir.
Now, the methyl transferases enzyme, methyl transferases. Now, actually, here the enzyme involved in, the second enzyme involved in, that is named guanyl transferase. This is second enzyme, guanyl transfer, remember. And another important one is, just wait, 1 minute. Right. So far 2 enzymes completed. One is triphosphatase enzyme. Second is guanyl transferase enzyme. Now the third enzyme is methyl transferase enzyme. Methyl transferase enzyme. The methyl transferase enzyme will add the methyl group at the seventh position of guanosine base. The methyl group varied from the adenosine. It is not important. Right. So remember one thing. The methyl group added seventh position of guanosine. Seventh position. This is the guanosine. In guanosine. A guanosine structure like this. It's a rough diagram. Here the carbon 1, 2, 3, 4, 5, 6. Some here the 7. The seventh position of the carbon. The methyl group added. Methyl group added. So the guanine, seventh group, methyl group added like this here. Methyl group added. So methyl group sometimes also added at 2-hydroxy H also. But most of the seventh position. Now capping protect 5' from the enzymatic degradation in the nucleus export the cytosol. Right. So okay, right. So the another important thing is the why the cap is required at the end of the 5'. Why the cap? The cap for the protection. Already this all reaction takes place inside the nucleus. In nucleus, number of exonucleases and nucleases enzymes are there. These nucleases enzymes will degrade the preparing mRNA. What happen if the mRNA degrade? Hello? What happen if the mRNA degrade? What happen? If there is no mRNA, no translation, no protein formed, no protein formation. To protect the mRNA strand, the 5 cap is required. 5' practice is required. Is it clear? Hello? Any doubt in this 5' cap?
No, sir, it's clear.
Right. But in our book, not clear. Please add this point.
Okay, sir.
Right. Let's see diagrammatically. So transcript, let's take pen, marker. This is transcript mRNA. At the 5 end, 3 primes, 3 phosphate, 1, 2, 3, 3 phosphate there. Next in this 1 phosphate removed. Phosphohydrolase enzyme, this first enzyme. From this one phosphate remove, only two phosphate there. Now guanosine transferases. In guanosine transferases, GTP triphosphate. In triphosphate also PP1. Two phosphate removed. Already told you. Two phosphate remains only one. Now this one added to the cap 5 end. Now methyl group added. Adomethane. That means methyl group added 7 methyl 1,7 methyl transferase enzyme. TEM7 GPP cap. Now this is completely cap formation, 5 end cap formation like this. This is the 5 end cap. So for the cap formation, the first phosphohydrolase enzyme involved. As a result, one phosphate removed from the triphosphate like this. And this is guanosine monophosphate. The guanosine monophosphate 5 prime, fifth phosphate attached to the 5 end. That's why this condition called as cap 5 prime to 5 prime. 5 prime to 5 prime. Because already 5 prime phosphate there. And remaining monophosphate also at the 5 prime. In guanosine monophosphate, the guanosine monophosphate at the 5 prime. Now the 5 prime end at 5 prime end. Actually, in normal attachment, always 5 prime attached to the 3 prime. This is normally occurs. The phosphorylation takes place between the 5 prime to 3 prime. But here, interestingly, 5 prime phosphorylation with 5 prime. That is very important thing. Right. I think it is clear.
Yes, sir.
Next one. Polyadenine tail. Uh, Polyadenine tail. Second one is polyadenine. Right. So this is also very, very important. A post-transcriptional RNA processing at the opposite end of the transcripts comes from the string adenine base, polyadenine base, end of the synthesized RNA chain 3 end, mainly 3 end. Already 5 end completed. In 5 end already, this is 3 end. 5 end already cap formed. This is 7-methyl guanine cap. This cap called as 7-methyl guanine cap at 5 end. So adenine is catalyzed by the enzyme poly-A polymerase. The mRNA first cleaved at 20 nucleotide downstream from AAUAA. This is the recognition sequence. Simple. Before polyadenine formation, what happened? Before polyadenine, there is a signal. There is a signal sequencer there. The signal sequences are at end of the 3 end. The signal sequences are A, A, U, and A like this. A cleavage sequence CA, GU rich. Sequence presents 20-40 nucleotide downstream to cleavage site. Simple. After this, let's change color. CA present. CA. This is the recognition site. CA present. After 20 to 40, here somewhere GU also present. GU. Guanine and uracil also present. right? So later another enzyme, poly A polymerase enzyme adds the poly A tail, which is subsequently extend the almost 100 to 200 adenine residues. Now the form poly A tail, which is presented three end and protected three end from the same nuclease enzyme like a cap. But what are the requirement? Here the formation of poly A, what are the requirement? Is there given any uh, just wait. Poly A signal, okay, right. Before formation poly A here, what happened? First poly A signal. So this is the poly A signal sequences after that, and as I said, CA sequence also present and followed by GU. This is poly A site. This is poly A site. Between CA and GU poly A site. Let's see how the poly A formation. Simple first. Some of the proteins are involved. And the first protein, cleavage, cleavage stimulating factor, cleavage stimulating factor, and another factor CPSF, cleavage polyadenine specific factor, cleavage polyadenine specific factor. Next, third one is PAPB, polyadenine binding protein. Polyadenine binding protein. These three important. Remember it. First one, CSF, cleavage stimulating factor. Cleavage, write down, cleavage stimulating factor. CPSF, cleavage polyadenine specific factor, and PAPB, polyadenine binding protein. Polyadenine binding protein. And another one also important here, the CF, cleavage factor. Cleavage factor. CF, cleavage factor, which act as a exonuclease. Like a exonuclease. What is the function of exonuclease? Hello? Exonuclease function is cut. Exonuclease function is cut. Right. So first of all, the CS sequence recognized by CSF. Later, the CS sequence from the right of the CS sequence with help of the CF cleavage factor. Cleavage factor, the cleave the phosphate groups here. Now it is separated. Now it is separated 5' to CA. Now the CPSF cleavage polyadenine specific factor add to this site and immediately added by PAPB, polyadenine binding protein. With help of the polyadenine binding protein, Now the continuation, adenine tail formed up to the 100 to 200 polyadenine tail formed. Polyadenine tail formed. Like this polyadenine tail formed at the end of the 3 prime. 5 prime already cap formed, already you knew that the later at 3 end polyadenine tail formed. So the function of polyadenine, so they are the two importance. One is for the protection from the exonucleus, from the nuclease enzyme in nucleus. Another one is it is act as a cleave. That means the separation of the mRNA from DNA strand. So these are the two important in polyadenine tail. Is it clear? Hello? Hello?
Yes, sir.
Any doubt in polyadenine formation? And guanine formation, any doubt? Guanine, 5-methyl guanine cap formation. 5-methyl guanine cap formation. Clear? Remember the polyadenine formation, the 3-end mRNA generated in 2 steps. First, endonuclease enzyme cuts primary transcript and appropriate location. Where is appropriate location? CA location. Here, CA. First of all, this CA location cut with help of the cleavage factor CF. And second step, poly A is added to the newly generated by enzyme called polyadenine polymerase binding protein. With help of the binding protein, the polyadenines are added, continuously added. Like this, adenines, polyadenine tail formed. So this is about polyadenine formation. But anyway, remember that only the simple two things are the first cleave and next add. Only two things. What is cleave? The cleave at specific site. The specific site CA. CA specific site cleaved. But net added with help of the PAPB, the adenines, number of adenines are added. Right. And what is the remaining? What is the next one? In this post-transcriptional changes, what is the next? Hello? In post-transcriptional changes, what is next? Hello? Already 2 steps are completed. How many steps in how many steps in post-transcriptional changes? Hello? Post-transcriptional changes steps. How many completed so far? Anita, Himabindu, Kankama, Hayan Sharif. Please respond. How many steps completed in post-transcriptional changes?
3, sir.
What are those? What are those?
Transcription.
What is that? Tell the answer whether it is right or wrong. What are the post-transcriptional changes? 3 changes. First one, 5-methyl guanine cap. Second one is polyadenine.
Yes, sir.
What is third one? Removal of introns, splicing. Splicing.
Yes, sir.
Now 5-methyl guanine cap completed. Second, polyadenine also completed. Now only one remains, that is the splicing. What is splicing?
Divisions.
Splicing means removal of introns.
Yes, sir.
Where is the introns? Introns the part of the RNAs.
Yes, sir.
After preparing mRNA strand from the DNA, in DNA actually exons or introns are present. But introns not code for any amino acids. That's why these introns are removed separately. Right. So cleavage polyadenine specific factor, okay, right, like this. If you see the diagrammatically, so this is polyadenine cap formation, right, like this. Cap, AO specific site, after cut the CA site and formation polyadenine, maybe the 100 to 200. Now let's see the splicing mechanism. Are you able to see the splicing mechanism slide, PPT? Hello?
Yes, sir.
Is it clear?
Yes, sir.
Right. Now splicing mechanism, for example, here the DNA, let's take DNA strand. DNA strand, exons, introns, exons. RNA prepared exons as it is transcribed, introns transcribed, exon transcribed. But this intron region not code for any amino acid. That's why need to be removed. Now the mRNA. But remember, don't forget already in this 5 cap formation, 5 methyl cap formation, polyadenine formation takes place. Polyadenine also formed. The next removal of introns. After removal of introns, here this red color, introns red color, and rest of the same. Now after remove exon 1, exon 2. Now this is called mature mRNA. Now this is called mature mRNA. From the mRNA, through translation, what is formed? Proteins formed. Yellow color protein formed. This is splicing mechanism. What are present in splicing mechanism? In this involved in spliceosomes or snRNA. Let's see the spliceosomes mechanism. First of all, this concept clear?
Yes, sir.
Right.
Yes, sir.
The sequence within the RNA determined with splicing occurs for 5 exons. Here the exon site. This is the introns. So here between introns and exons, let's take spin. Draw this. Exon. For this exon G. Here exon G. Guanine. GU guanine uracil. Adenine guanine. End of the introns. This in introns start with GU, end with AG. GU, AG. Now this is the branch site. Remember, this is the A is the branch site. First of all, remember this. What happened here? The spliceosomes, a set of small RNA molecules called snRNAs. The 5 snRNAs called U1, U1, U2, U4, U5, U6, U6. These are all called, this group called as spliceosome. A spliceosome performs the process of RNA splicing in the following way. First, First, first step, the splicing junction GU. This is splicing junction GU, recognized by U1, splicing junction, recognize U1, snRNA. Second step, the right splicing junction is recognized by UF. This is the right second AG. This is recognized by UF. Third, branch site, the consensus sequence recognized by U2. The branch, this is branch site consequence sequence recognized by U2. And U4, U6 function so far unknown. U4, U6 with U1, U2, U5, the causing the entrance loop. Now the maybe the remaining formation, the entrance loop, formation entrance loop. The spliceosome also catalyzes the cutting and joining the reaction that excites the entrance and ligase exons. After that, with help of this splice, the after remaining all formed, remaining three also formed, the somewhere attached. Now it is cut here and also cut here. That is the function of nucleosome. Later, this entire position separated. Now only the blue colors, these green colors are attached. These two colors attached. Now this light color, this entrant region is removed. So this is done by spliceosome. This spliceosome. Is it clear? Hello? Hello?
Clear,
Yes,
sir.
sir.
Right. The next
Yes,
one.
sir.
Right. You can see diagrammatically here. Entrance lariat. That means one end called lariat. The spliced exons. Pi cap site. This is the branch point. 3 site. So this chemical structure noted, but remember this part. Exons entrance. This sequence separated and joined these two. Or you can write the simple exons, entrance, exons, remove the entrance and both exons joined. The very simple. This is the spliceosome mechanism. This entrance removed. Now this exon. This is important. This diagrams required. The simple exons between entrance. Entrance with spliceosome. These are the spliceosome. These spliceosomes the fold the entrance. Now cut the entrance and separated. Now join. This is the simple diagram. Draw this for spliceosome. Right? Is it clear?
Yes.
Take this picture.
Yes, sir.
Right. So now these three completed. Right? Any doubt in these three aspects?
No, sir.
Right. Now the matured RNA prepared. After that, this mature RNA perform the next. What is the next stage?
Translation, sir, for protein
Translation.
synthesis.
Right. Translation for protein synthesis. After the formation, this mature RNA now transported into the cytosol, where the translations takes place. Right. Now once again, recap this first transcription process to post transcription process. First of all, in eukaryotic transcription, how many stages? First of all, what is transcription? Tell me. This is very, very important. This question important. Tell
Synthesis
me.
of mRNA from the DNA template.
Right. mRNA from the DNA. That is called transcription. This transcription process takes place inside the nucleus. But what actually, while the preparing RNA, the required some of the unwanted genes also transcribed in form of introns. And this RNA, as it present inside the nucleus, this nucleus position dangerous to RNA because in nucleus some nucleases enzymes are there. This nucleases enzyme kills the RNA. To protect this, always remember the nucleases enzyme always attacks an end of the strand, not middle. End of the strand. That's why the both ends need to be protected. The both ends of the hnRNA, premature RNA, need to be protected. That first 5 prime with protected with 5 prime protected with, hello, how the 5 prime protected in
With
mRNA strand? How? With,
methylated capsule.
right, 5 7 methyl guanosine cap. That is first one. And second one, and that means tail. Which tail?
Poly A tail.
Polyadenine tail. After polyadenine tail, next the introns also removed in the form of, in this process of plascisome
Lysosome.
or splicing. Splicing
Side.
process in this involved the spliceosomes. These are the spliceosome U1, U2, U3, so U4 and U5, 6. These are the spliceosomes. With help of the spliceosomes, the introns are removed. Right. So now the matured RNA prepared. This matured RNA translocated to the cytosol where the translation takes place. Now what is the translation definition?
Synthesis of protein from matured RNA.
Right. So there's a simple way, genetic information in the form of codons decoded or translocated in amino acid to polypeptide chain. This process is called translation. So the simple definition, as you said, that is protein formation on the mRNA strand. That is also right. Protein formation or amino acid chain, polypeptide chain formation on the mature RNA. That is called translation. Now the translation, let's see the some of the requirement. Let's take PPT. The way it is protein eukaryotics. Slide, PPT slide visible or not?
Yes, sir, it is visible.
Right. Here prokaryotes, no, not in prokaryotes. We need right, eukaryotic translation. Right. So first of all, let's see the requirements of the translation. Let's see the requirement. Just write down one by one. Or what are the requirement for the eukaryotic translation? Mature mRNA, first one. Hello? Are you follow me?
Yes, sir.
Yes, sir.
First requirement is mature mRNA. Already we get that, right? Second, transfer RNA, which are present in cytosol. Third, ATS ribosomes. ATS ribosomes. Fourth, write down all amino acids, which is also present in cytosol. And some factors. Factors. Factors means initiation factor, elongation factor, termination factor. And in translation also, like a transcription, three stages. What are the three stages? What are the three stages in transcription? Hello? Transcription stages. Hello? Voice clear?
Yes, sir.
Tell me
Yes, sir.
transcription changes. What are the transcriptional changes? Eukaryotic transcription stages.
Sir, initiation, elongation, and termination.
In order. All are right, but tell me in order.
Initiation, sir, first.
Right, first initiation,
Then elongation, termination.
second elongation, third termination. The same translation also, three stages. First initiation, second elongation,
Elongation.
third
Chain
termination. Third termination. Right? And before the initiation, some of the stages also there in eukaryotic translation. That is, first of all, you know what are the requirement for the translation. This is first step. These are all requirement. The second, next one is activation of amino acid in tRNA. This is the part of initiation. Actually, okay, let me complete. The actual first stage is initiation. Second stage is elongation, and third one termination. First, let's complete initiation. In initiation stage, first required the activation of amino acid. Let's see how the amino acids are activated. First, in initiation process, first amino acids are need to be activated. For amino acid activated, first here there are 20 different types of amino acid present in cytoplasm, present in the cytosol, not plasm, cytosol. They are all inactive state, in which are present inside the cytosol, 20 types, but they are not active. They need to be, they need to be active. So now this inactive 20 types site, which activated with help of the ATP aminal acyl, ATP aminal acyl synthase enzyme, with help of ATP by the enzyme name is amino acyl. Write down all this. Amino acyl synthetase. Amino acyl synthetase. Amino acyl synthetase. With help of ATP and amino acyl synthetase, these inactivated enzymes, inactivated amino acids are activated. This is the activated. Right? Is it clear? In initiation, first step clear? Hello?
Yes, sir.
Right. Now this activated amino acids attached to tRNA, CC end. As a result, tRNA activated. That means now this activated amino acid attached to the amino acid. Activated amino acid plus tRNA. One complex formed. This complex called as ternary complex. Ternary. Write down all this. Ternary complex. Ternary complex. One minute. Ternary complex. This is second step. So ternary complex also formation. And third step, 43S initiation complex formation. Second, 48 initiation complex. Third, final 8S initiation complex. First of all, in initiation, in first step, what is activation of amino acid? Inactivated amino acid activated with help of the ATP amino acyl synthetase. That is first step. The second, ternary complex form. Ternary complex means amino acid and with tRNA, add with tRNA. Now this is called ternary complex. After that, 43 initiation complex formed. 43 initiation complex. Now this two clear. First of all, hello?
Yes, sir. Clear, sir.
In initiation process, first activation amino acid, next ternary complex formed. Is it clear? Two clear?
Yes, sir.
Yes.
Yes, sir.
And third write down in rough. Third 43S initiation complex. Fourth 48S. One is steps 48S. Final 80S. These are the step one by one. Did you write 43S, 48, 80S? Hello?
Yes, sir.
Right. Now in this actually so far two completed. One step is activated amino acid completed. Second one ternary complex formation completed. Clear? Hello?
Clear, sir.
Right. Now 43S complex formation. Let's see 43S. Right here, write down this formation of 43S pre-initiation complex, 48S initiation complex, 8S initiation complex. Before that, you should write what are the two remaining two. Actually, not given here. What are the remaining two? Hello? In initiation, five aspects mainly. 43, 48, 8. Before that, what are the two completed already? The two completed, what are those? Hello? Please focus on the topic. Please listen, class. Hello?
Yes.
In initiation, what is the first step?
mRNA.
Not I am asking requirement. I am not asking about requirement. They are the requirement. mRNA requirement, amino acid requirement, ribosome requirement. They are the requirements of the translation process. But I'm asking what are the initiation steps? In
Sir,
initiation
first thing
first,
is activation of amino acid.
right? Second one,
Then ternary complex formation. That is
right.
activated amino
Second,
acid is bind to the tRNA.
that's it. Next 43S, 48. Now let's discuss about 43S complex formation. Right. The formation 43 pre-initiation complex. Now the ternary complex. Already you know the what is ternary complex?
Yes, sir. Amino acid with tRNA.
Right,
Activated amino
right,
acid.
right. So this ternary complex that is containing met tRNA, and there are the another factors involved in this IF2, the bound to GTP attached to 40S complex subunit to form 43 pre-initiation complex. Simple. Uh, this is tRNA structure. tRNA CC end. The CC end attached by methionine amino acid, that we met. Activated methionine amino acid. First amino acid, that is called met. Methionine, met for methionine. So now this activated amino acid attached to CC end. Now this is called ternary complex, right? Right?
Yes, sir.
Now 40S ribosome in the eukaryote 40S and 68. 40S ribosome. Here. This is 40S ribosome. Now what happened? And EF2 bound to GTP attached to 40S ribosome subunit to form complex. Now, not here. 40S here. Just wait. This is 40S. 40S subunits IF3. IF2. IF1A. This all attached to the 43S. 40S subunits. This 40 subunits, after attachment these factors, now it is called 43S initiation complex. 43S initiation complex. Now the both attached. Now the both means in 48S, in 48S, this is ternary complex. Ternary complex. This is 43S, right? Now this both ternary and 43S both attached and formed as a 48S. Is it clear? That is simple diagram. 40S the tRNA. tRNA amino acid. tRNA amino acid. This one, tRNA amino acid. Now this is called 48S. Is it clear first? Hello? Hello?
Yes, sir. Yes, sir.
Ternary complex completed. This ternary complex. Now this is 43S. That means only 40 subunits. It made up of 40 subunit with the for some of the factors. Now this 43S. 43S plus ternary forms the 48. Simple. Form the 48. Is it clear?
Yes, sir.
Right. Now the final one, what is the final? After 48,
ATS, ATS.
80s. The simple 60s subunit attached to the 48. This is 48 and attached by 60 subunit. This is 60 subunit attached by 60 subunits. That is called ATS, which is with help of IF1. This subunit attached with help of IF1 initiation factors, IF5, sorry, initiation factor 5. Now clear? ATS also clear?
Yes, sir.
Right. Let's see diagrammatically one by one. Sure, not given. Actually, diagrams not given. Give the diagrams. Just wait. Is it visible? Hello?
Yes, sir, visible.
Are you able to see 43S complex? Here.
Yes, sir.
This is 40S subunit. This is tertiary tRNA met. This one tRNA. When the both attached, this is called 48S actually. Right? And small in it, the after finally large subunit added like this. Now this is ATS, right? Hello?
Yes, sir.
Right. In this ribosome, the another important thing is after formation ATS ribosome, uh, the three important regions present in this ribosome, E, P site, A site. Listen carefully, very, very important here. Without understanding about these sites, you can't understand translation process. Remember. So E for exit site, P for peptide site, A for amino acid site. Right? Clear?
Clear, sir.
Right. Now let's see the elongation process. In elongation process, Premature complex, okay. Where is diagram? Right. What is elongation? In elongation, ribosomes elongated the polypeptide chain by sequential addition of amino acid. That is the important. Sequential changes, the simple, by adding of amino acids. The amino acid sequence is determined by the order of codon in the specific mRNA. The specific mRNA, for example, this is mRNA. mRNA already triplet codons are there. This triplet codon, specific RNA. Elongation cycle process involving certain elongation factors. Like initiation factor, some are elongation factors also involved. EF, IF initiation factor, EF elongation factors. So elongation may be divided into 3 steps. They are binding amino acid tRNA to A site. Important. Peptide bond formation, translocation. Is it clear? 3? Hello?
Yes, sir.
Already told you A site and P site, E site. In this first, in elongation process, amino acid tRNA bind at A site. Next peptide bond formation between amino acid. Next translocation. Peptide bond transformation. Let's see where is diagram. Right. So elongation cycle, see here. This is 40th subunit, 60th subunit, E site. Are you able to see E site? Is it clearly visible or not? E
Yes,
site,
sir.
P peptide, A site. Before elongation, in the end of the initiation, actually already the met tRNA, first tRNA, methionine tRNA present at P site, peptide site. At the peptide site, already one tRNA present. One tRNA present. Now, what is the free site? Free site is the A site, amino site. The amino site is free, but peptide site already tRNA present. Before elongation, what happened? The incoming amino acid tRNA elongation factor 1 with help of elongation factor 1. Elongation factor 1 reached toward the A site. Reached to the A site with help of elongation factor 1. And also in this involved ATP energy. Right. Now, where is second tRNA? Now, this is the first tRNA amino acid. The red color is the second tRNA amino acid here. The pink color, second tRNA amino acid. So, the first is clear. Remember in this process, first of all, the first tRNA amino acid binded peptide site, P site. But the next coming, the upcoming next one, all amino acid attached at A site and shifted to P site. It is continuously happened. A site to P site, A site to P site, right? So next step, second step, peptide bond formation. How the peptide bond formation actually, if you see this arrow mark here, now this first tRNA, this is second tRNA. The bond formation between first tRNA amino acid to second amino acid. Where is first amino acid? In this green or pink, where is first amino acid? Hello?
Green, sir.
Green first amino acid. Where is second amino acid?
On pink one.
Pink one second amino acid. In between what happened? The peptide bond formation. Peptide bond for I will show later how the bond formation between one amino acid to the another amino acid. So anyway, the peptide bond formation between one amino acid to the another amino acids. Now that is the second step. Peptide bond formation with help of peptidyl transferase enzyme. Now the second step also complete. Peptide bond formation. Now the third step is translocation. The translocation means the first tRNA separated. That means translocate the first tRNA, green one, first one. Now the first tRNA separated. First tRNA separated. Now later the A site tRNA shifted to the P site. A site tRNA shifted to P site. Now what happened? The P site tRNA? P site tRNA reach E site, exit site. From exit site it is exit out. Hello?
Yes, sir. Understanding.
Right. Now which one is free? Which site is free now? Here.
Yes, sir.
Right. Now the coming all amino acid come to the A site and shifted to the P site, translocated to P. A site translocate to P. A site translocated to P after peptide bond formation. After peptide bond formation. This is continued until nonsense codon reach. Until nonsense codon reach at A site. Until nonsense codon reach at A site, this will be continued. When the A site reached with nonsense codon, now it is terminated. When it is reached, now it is terminated. Elongation clear? Hello?
Clear, sir.
Right. Termination, when ribosome unit reach the nonsense codon, the process is terminated. In this termination, with help of release factor 1, release factor 2, and GTP. Let's see the termination process. In the nonsense codons UGA, UAA, UGA, these are nonsense codon, or stops codon. When it is reached, release factor 1 recognize all the three nonsense codon. This RF3 release factor 3 stimulate the termination event recognized by F1 release factor 1 stimulated by F3. Now automatically what happened? When these codons reach the A site, UAG, one of the nonsense codon, it is recognized by release factor 1. and it is RF3 stimulated. Stimulated means now the peptide polypeptide bond separated. Separated. Now the proteins form. Now the protein is formed. Now this is called as protein synthesis, three steps: termination, elongation, and initiation. Now let's see the simple process here. For example, this is mRNA codons. That 40S here already T first amino acid T. This is P site, A site. This is E site. tRNA second. So what happened at the first amino acid? Bond formation to the second amino acid. Later it is shifted. This tRNA shifted to P site. This translocate to A site. The next what happened? P site common, A site common. Already amino acid two already here. Peptide bond. First amino acid one. First this is first amino acid. Peptide bond with second amino acid. Now what is third amino acid coming to the A site? Same peptide bond formation like 1, 3, 2, 1. Now this keep going continuation. Peptide bond. Here everyone peptide bond formation. A peptide bond formation. Like this peptide continuation growing chain called as protein. Like this. This is 1, 2, 3, amino acid 4, 5, 6. Like this. After formation, when reach the UAG code with help of release factor 3, it is cut the protein strand. Protein strand. Right? Is it clear? Hello?
Clear, sir.
Right.
Sir,
Now, tell me. Hello? Hello? Tell me, what is your doubt?
Sir, nothing, sir. Everything is clear.
Right. Let's see the bond formation between one amino acid to the another amino acid. But but it's not requirement, but our clear understanding I will draw. Just wait. Amino acid for example here. Amino acid structure 1 C H N H2 amino acid. This is amino group. This NH2. This is NH NH NH2. And here alkyl group. C keto OH. This is amino acid structure. And the group of amino acid based on this alkyl group. Right. Plus another one is, this is carboxyl group. Another amino acid, same NH2, NH2, COR, C hydroxy group, alkyl group, keto, which. So actually the bond formation between carboxylic group of the first amino acid and amino group of the second amino acid. Carboxyl group and amino group. Here from the carboxyl group, one AH hydroxyl, and here one hydrogen loss in the form of water molecule. Form of water molecules. Now the bond formed. That means there is no OH. There is no OH. There is no H. Now here only NH. The bond formation between keto COO. Let's change color. Already OH separated. And here this is peptide bond. The red color mark is peptide bond. So one peptide bond formation between carboxylic group and amino group. Is it clear? Like this, the every bond formation between one amino acid to another. These bonds number of, that's why called as a polypeptide or polypeptide chain. Is it clear? Hello?
Clear, sir.
Right. But in examination point of view, no need. Just write the bond formation, but remember it. Okay? Any doubts today class?
No, sir.
Right. Can I quit?
Sir, one doubt. Like post-transcriptional modifications, is there any modifications occur in protein formation, sir?
That means after formation protein or before?
After formation of proteins,
Hmm.
is there any modifications take place like
After protein formation also there is post changes are there.
okay, sir.
I will explain later.
Okay, sir. Okay, sir.